Well, thank you very much, Mark, for the invitation, and thank you everyone for being here for nice talks we just heard. And it's nice to see some friends that we're working also on tyrosine phosphatases. So I was not sure what I was supposed to present. I don't want to just show you a company. I think much more important is the work we did for 30 years on tyrosine phosphatase. And every time I mentioned that to my department, they all laugh about phosphatase. They didn't want to hear about anything. So I'll give you a bit of phosphatase 101 and to explain to you where we are. So I think I can just click on this. Yeah. So I'll go back far. So this is Dr. Rouse that end up studying an infection, he thought, initially in this chicken, in the Plymouth chicken. And so they had big tumor. In fact, it was a tumor. And this can be transplanted to other chicken. So eventually, many years after, other people, Howard Temin, for example, that also discovered reverse transcriptase, identified that this was a virus. They have an assay for this. So there was something in these tumors that was causing cancer. So we're going fast to get to the 10 minutes. So what happened is that this virus was identified as the Rouse sarcoma virus, was named as his name. And many people worked for about 30 years trying to identify each genes and which genes was causing cancer in this virus and the chicken. And one of them, as you can see, is SARC, the green little box here. And this is a SARC kinase. So it's a tyrosine kinase specifically that phosphorylate protein on tyrosine. And by doing this, you cause cancer in the cells that you transform the cells. And this is work by Tony Hunter that in this paper where he identified the SARC kinase to be a tyrosine kinase. And then you can see here, we knew a lot of phosphoserine, phosphothreonine, but this one, if you overexpress SARC, you get a phosphotyrosine amino acid. So this from the chicken to the identification took about 60 years. So, but it's a remarkable and like many stories in science where you have increment growth, and every single year that pass and people put their bricks in and you build a house. So if you go back to human kinome now, we have quite a lot of tyrosine kinase, but they all come from the same ancestor gene. So it is well known that all the kinases that were made, serine, threonine, and tyrosine, they all come from the initial gene that shows up. As a PS, by the way, the tyrosine phosphatase show up in evolution before the kinase. I'd like to say that, but you can tell me why later on. But that's the case. So there's still quite a lot of tyrosine kinases, and in about we're now in 24, so in the 1980s. When we started identifying oncogene, most of the oncogene were tyrosine kinases, and some of those, BCR-ABL, RAF, PTK, but RAF is a different type, EGF receptor, JAK kinase. So clearly, this is at the time I arrived in my graduate studies, and I was working on adenovirus at the time. I liked viruses. I thought it was very cool. But then it was time for me to learn English. I was in Sherbrooke. So I decided to move to McMaster University to work with Phil Branton. Not only I wanted to work with somebody that was doing something exciting on tyrosine phosphatase, was trying to identify them, but he was a wonderful person. As you know, he passed away this year. So I arrived in his lab, and there were several things they were doing in the lab. So they were trying first to use SARC from the virus itself, and then they were infecting chicken embryo fibroblasts and putting radioisotope in it, gamma ATP, that label protein. So you can use this as a substrate for trying to find. And we had an incredible large amount of radioisotope in the lab. We were the only biology lab with forceps screen anyway. So I got a lot of irradiations. Today, to this date, the entire radiations of the Goodman Cancer Center is about 10 times less than my own. So it was interesting. But I certainly learned about tyrosine phosphatase in Phil's lab, and he was the first paper published in MCB on a peak of tyrosine phosphatase. He thought they had that, but finally at the end, it was a bit more difficult than he thought. At the same time, I was finishing my PhD on tumor viruses and also phosphorylation. There was all this work on starting on embryonic stem cells, trying to see, can we mutate genes in embryonic stem cells and then create animals which will be mutated. Today, it's easy. You get the CRISPR-Cas and you do it. In those days, it took quite a long time. So in fact, these gentlemen, Mario Capecchi, Martin Evans, work on the stem cells itself to make embryo. Oliver Smithies work on homologous recombination. So they got the Nobel Prize together, but none of these guys worked together. They were really independent at the time. But Oliver Smithies was a colleague of Kanyr Westphal that I work with at the NIH. And so we made the first animal which had a phenotype. Smithies got the Nobel Prize. He got HPRT, but HPRT in mice has no phenotype. There's a salvage pathway for Lesch-Nyhan disease. So they didn't claim, they claimed the mutation for the first time. But I must say that Oliver Smithies helped me personally and the group in the lab. So we published this. This is Gaucher disease. That was the first animal. And at NIH at the time, there were about 60 kids trying to treat these kids with proteins that we're making. Each kid had a different concoctions by the companies that was working at NIH too. So finally, this model was what allowed them to try to test many different animals with many different drugs. And now there is it's startup for some of the key protein that you can get for Gaucher disease. So that was my little things. But at the same time that I was using this technology and learning about it, then Nick Tonks published the first tyrosine phosphatase. So there were one. And the reason why I was interested immediately is because of Phil Brenton's work. And it makes sense that if you have a lot of oncogenes that are tyrosine kinase, you would expect that the phosphatase will do something, either make it worse or finding a way to do it. So when he published his first enzyme was PTP1B, it was a second peak. And they find out that CD45, that many of you may in immunology use CD45, well, it's a tyrosine phosphatase, it has a very important function. So you cannot say you're not working on phosphatase. Well, I just want to say that again. I feel like I have to defend myself even years after 33 years. So anyway, so this was my plan. My plan was not a great scientific ideas, was to use knockout and try to develop use knockout and try to develop a mutant mouse for tyrosine phosphatase. Now there are 107 of those. And I had cloned many before leaving the NIH. I had cloned a good 20 to 30 myself, new ones. And 80% of the 107 are in immune cells. So it turns out that we started to work on all of those, and Yuri works on the receptor phosphatase sigma. It's a fantastic one for nerve regeneration and other things. And PTP1B and T-cell phosphatase, that's the one you see here. There are two different enzymes, but very similar. But in fact, in Drosophila, it's the same gene. So, it made sense to me to work on genes family, subfamily, because they could compensate for one or the other. So we start working on this one. The T-cell phosphatase was my first grant at and then the NCIC. My first operating grant when I arrived at McGill, and I'm still working on it. So that's some of the things I'm doing. So we made the knockout of PTPN2, T-cell phosphatase, PTP1B. We mated them together. And this was important because at that time we show the phenotype was increased in terms of creating a signaling from JAK-STAT signaling. So it is much better to inhibit both of them than only one. And indeed, the Drosophila, so Norbert Perrimon did this work. He wanted to find which gene can you use siRNA to which one of the genes of Drosophila will create the most effect on JAK-STAT signaling. And it turns out it's a gene called PTP61F. And there's two alternative splice forms. And these alternative splice forms exactly look like 1B. And so they diverged during evolution, and it made sense to study both of them. So the knockout that we had on the T cell phosphatase increased if there was a severe inflammation signal. So the mouse died very quickly, about 4 weeks, 4 weeks after it's born, and it was you cannot study more than the cell itself. You tell me when who has the. So I'm going to look at this way. But anyway, so sorry. And so we did all this, and we work, and I can say to you that we at that time we published a paper in Science with Merck Frost. Merck was very interested on PTP-N1, so PTP-1B, because there was several data in the literature suggesting that this enzyme was higher in type 2 diabetes. So, and also Pima Indians, the Inuits in the north, if they were obese, they had lots of this gene. So if you knock it out, you could probably treat diabetes. So they had a gene, they had a molecule that they developed, and all the inhibitors of these two genes that I will tell you are based on this idea that there's two binding pockets and not only one. And so you can make small molecule, and these are the Merck Frost 598, Connect, and the one we use at Kanyr is I'll tell you about Kanyr later. KQ791 has two pockets to inhibit, and so therefore it's extremely specific for these two. So it's a small inhibitor, and it does not touch any of the other tyrosine phosphatase. So Merck went to Connect because Merck closed in Montreal. They give all of these compounds to ex-Merck Frost people, and they did in clinical trials. The first one was very nice, very stable. They could inhibit relatively well, but it was just the safety, tolerability, pharmacokinetics. And there's a lot of great story on this compound. They did also a phase 2, and turns out half of the patients with type 2 diabetes had a reduction. The other half did not at all. So the FDA told them, I'm sorry, but you have to go back, repeat this, and find the difference between those that respond, those that don't respond. So, but then at that time, our Quebec venture that was funding them decided to quit. They didn't have the money for that. So the company was shut down, and all the IP came to the same three guys that developed this, but with a company that we started together, the four of us. And but during that time they were doing all this, the patent was evolving, of course, that years are passing, and my gray hair showed up, all the four of us, in fact. And on top of that, what happened is that we were continuing working in the lab producing paper. We have over 100 papers on these two enzymes. So we're very, we knew that. These are some of the papers we have. I say 90 papers, but I say over 90, not in that neighborhood. And so we had very nice papers showing that this could be used in immunotherapy. So if you can do this to a mouse, you can inject the compound, and then after 20 minutes, you can see the rise of phosphostats. Phosphostat 3 is not necessarily bad, by the way, in immunotherapy. But so we could see and follow that. And therefore, we move on this, and it's now a compound. KQ791 is a compound for Kanyr. Kanyr is snow in Inuit. Kanyr is just the tyrosine that I just thought about. I could have taken. My other company is called Mispro, so that's even worse as a name. So I'm not very good at that. But the compound works very well. We had we got, and this is a point that we can discuss. The Quebec government with CQDM, the other group of funding from the government can help you go forward. This is a test on mice for breast cancer that was totally funded by the CQDM. And so we don't have so much money. The drug is available in pills, and we're continuing doing work on that on many different cancer types. So in fact, worse than that, or better than that, we also examine for each cell type. We make a knockout in dendritic cells only, and T cells, and CAR. So we were able to check by genetic means and also by the drug, all these different, and we end up with a GAP program, a genomic program to develop a cord blood NK Bank at MUHC with a $5.2 million for the bank to identify cord blood NK cells that we can activate with our compound. And we do see now quite a variety of cells responding. We're now looking at the genetic of those cells. Which cells can we have a signature motif that will give us activations by the compound, or even that if they are themselves outstandingly good to kill tumors, that would be for the most, I'm almost done. Time is up. So, but most important, we were looking for using the pills to patient, and that's what we were trying to do. But I'll finish by saying the times is sometimes difficult. Here is all good things. In the bottom here, composition matter, there's 5 years left. So it's very difficult for a company to fund you. So we're making derivative. And then this paper came out because now our work was starting to be noted in many places. We publish in many nice journals. So this is a paper with 52 people. And this is AbbVie that has inhibitor, and they repeated mainly the things we have, but in one paper. And they put 40 million in that, if not more. And on top of that, what did they do? Well, that's what I have to finish, so I stop. And now tyrosine phosphatase is one of the PTP1 and N2 is one of the very interesting target for company. Genentech, Anavo, Kanyr is our company. Boehringer Ingelheim just bought a small company for $1.2 billion for their compound on T-cell phosphatase. So I'm very happy with that. I think I can die with saying that our paper is okay. I don't expect, I made the one with the Miss Pro, I made my money out of that. And I'm very interested now to see if we will be able to get to the end of this place. Okay, to push the compound in there. If it's not us, the big pharma, if the big pharma don't succeed, well, by as well, that means we would never succeed. So that's it. It was fun talking to you, and I'll answer questions.